All right. Hey, we're going to turn it over to our featured speaker. We're very fortunate tonight to have Dr. Jennifer Tsuruda. Uh, oh, she's coming. She's fixing to come on board here. I seen her pop her face up right there. So we're excited to have Dr. Tsuruda with us tonight. She's been on board with UT since February of 2019 as our beekeeping specialist. She's when I look over her education and stuff, looks like she got all of her BS, MS, or PhD and all that stuff from University of California, Davis. And then she went on to do some research work at Purdue. She was the apiculture specialist at Clemson, and now she's here with us in Tennessee. She's very informative about bees and beekeeping, and so we're excited tonight to learn about murder mites, as she calls them. So turn it over to Dr. Tsuruda.
Well, thank you so much. Let me try sharing my screen. Hopefully we'll have no technical difficulties, but I do have some videos to try to keep everyone engaged and awake, but those can sometimes lead to some issues. So Chris just put up a poll. Thank you so much, Chris, for getting those put together. So just a little pre-poll, and it's okay if you don't know the answers because we'll do the post-poll afterwards, and that's where the answers I expect or the percentages to be more correct. But for now, please go ahead and fill out that pre-poll just so we get a sense of where you guys are and your thinking. And I'll start sharing my screen. So I was really interested to see how many newbies, as we call it, are on the call. So some of this material may be a little bit out of context for you until you actually have bees, but I think it'll still be a value for you to understand some of the challenges that you'll face when you get into beekeeping. But yeah, I guess I expected more of a crowd that had bees already, but that's okay. We'll just make sure that we have time for questions at the end for you all to ask things. If we need to go back to some really basic level information, then we can go ahead and do that. So don't worry. And so sorry, yeah, you can see I have a no photograph icon on like all of my slides because yeah, I tend not to do recordings because the information gets out of date rather quickly, and also I run the master beekeeping program, and so I try to keep my content somewhat in control, or I try to keep control of my content, I would say. Okay, so we have about 77, we're missing about 6 people from filling out the poll. So if you're out there, please fill it out. But yeah, tonight I'm going to be talking about what I'm calling murder mites. And they're Varroa mites. It's the same old Varroa mites that we've been dealing with for, well, a little over 30 years now, but trying to put a different spin on it, because I think almost like with COVID, people are tired of hearing about Varroa mites, and sometimes they lose motivation to try to control and manage them. And I'm going to try to make the point this evening that management is the key for keeping your bees healthy, surviving, and then thriving, because we don't just want them to stay stagnant. We really want you to be productive and have your colonies grow. I guess we'll just go ahead, Chris, if that's okay with you. I mean, we don't have everyone filling out the poll, but I just don't want to wait too much longer since we're almost 10 minutes in. We will go ahead now and just kind of giving you a little bit of background is, you know, part of my job is dealing with bees and beekeepers, but also I kind of get calls about everything related to insects. And so obviously one of the big ones this year was the Asian giant hornet, which the acronym is just appropriately, So these buggers, you know, they're not native to North America. They're from Asia. We have this issue with them popping up in British Columbia and in Washington State. And of course, they have a really bad reputation for not only being a major predator of honeybees, but also in their native land, actually contributing to some human death. But I would also argue that there are plenty of honeybees that also contribute to human death as well. So this is why we call them the Asian giant hornets. We don't call them murder hornets, but they were, whoever came up with that nickname moniker was really smart. And so that's why I'm going to try and build upon that to talk about mites, because really these Asian giant hornets, at least, especially where we are in Tennessee, These are not a problem for us. Varroa mites are definitely a problem for us. We are losing colonies in Tennessee to varroa mites. We are not losing colonies to Asian giant hornets where we are. So, it did lead to a lot of cute little memes that were out there and, kind of related to COVID. And it gave us a little bit of sense of um, humor, but also just kind of putting things into a reality check. But I just also want to make sure that you all realize and are aware that these are just some of the samples that got sent to me for identification, and none of these are all from Tennessee. None of these are Asian giant hornets. So don't worry about that. I just wanted to make sure that I didn't give you the impression that we have them here. We do not have them documented in Tennessee. So far in the United States, they've only been documented up in Washington state. So we're not going to worry about those right now. The bigger pressing issue that we have with honeybees are these varroa mites. They're also from Asia, from the Asian honeybee, and they made a host switch onto European honeybees. And decades ago, we were a lot more loose with our trade and moving bees around the world and even around the United States. And so they made a host switch. And then when we moved Apis mellifera, which is our European honeybees, around, we ended up getting these mites here. And so we've been dealing with them for over 30 years now, and they're still very much a problem. They are contributing to very high losses. Since there are so many of you who are new to bees on the call, I'll just point out that before I got here, I think the year before and maybe 3 years before I got here, Tennessee had hive losses that were over 70%, 70%. And you can imagine that's not obviously not sustainable. And there are a lot of things that can lead to losses of colonies, but one of the big ones are varroa mites, and that they're an international problem. But part of how we manage them in the United States leads to them being a bigger problem here. We're moving bees around a lot. Um, our management practices are not always very efficient, and we're learning more. I mean, that's one of the problems, is that we're learning more as we go along. So we do have definitely room for improvement, and we'll get to that in just a second. But I just want to point out a little bit about these mites. So if you haven't picked them out yet from this pic for the video, they are these reddish brown discs that you see on these bees, and they can lie very flat on the bees. And they feed on developing bees and adult bees. They have a really tight relationship with bees. And so I'm just going to very briefly go through this life cycle of the varroa mites. And you can tell by this picture, they've been studied for a very long time. That these drawings are rather old, but they still hold true to the biology. And so we start up here with this bee number one. This is an adult bee, and it has a mite on her. And those mites are very in tuned with reproducing and being successful. So they will then jump off that bee and then enter a developing brood cell, where we call developing bees brood. And the early stages are larvae, and then after that they pupate and turn into pupa, which is like the chrysalis form for a butterfly. But these mites are really good about sniffing out and knowing when there's a brood cell that has a larva that's about to pupate. So they enter in this uncapped cell, so an open cell, and they go down and they hide in the brood food. That provides them a little bit of protection, and they can hang out in there, and they go into a little bit of like a diapause kind of a winterish kind of period, or just kind of a um, calm period where they aren't doing anything but just mellowing and hanging out there until they get the stimulus or the trigger to then start feeding. And so once that cell gets capped off, so when the bee becomes of age to then start pupating, the older adult bees will then cap off the cell so they can finish their development in a closed cell, just like a chrysalis, that magical metamorphosis from a caterpillar looking individual to an adult happens all undercover. So once that cell gets capped, one of the little mite will then come out of the brood food, and it'll start feeding on this developing bee. So Scott, I don't know if can you control the participants? There's someone's not muted. Just wanted to let you know. So that bee or that mite then starts feeding on this developing prepupa. The bee starts pupating, and then this mite, she's actually already been mated. So she starts laying eggs on this developing bee. So first she lays a male egg, and then she starts laying female eggs after that. And then those eggs hatch, and then those mites start developing as well. Once they become old enough, they will reproduce in the cell. So there's a high level of inbreeding that happens with these mites. But you can have multiple mites enter a cell as well. So you can have some outbreeding as well, but it's a pretty inbred system. So now the daughter mites and the foundress mite that started this all off are mated. And then once that bee emerges from that cell, it's completed its development. Those mites then leave too with that bee. And then bees are living in such close proximity to one another. They are constantly interacting with one another. It's very easy for a mite to transfer from one bee to another bee. And that takes us around to the start of the life cycle again. And so really when we talk about the life cycle of these mites, we have the dispersal phase, or sometimes we call it the phoretic stage. And that's when those mites are on the adult bees. But then we also have this reproductive phase, and that's when they're inside those developing brood cells reproducing themselves. So what this looks like in reality here here's a nice little family of mites in here. And so you can see towards the top, this reddish brown individual. So that's the foundress mite. And these are the daughters, and the son is in there as well. So even though it looks like there are a lot of daughter mites in here, given the development time of the mites, only about 1 to 2 of these daughter mites will actually mature and be mated. So even though it looks like a scary number here of offspring, you only get about 1 to 2 daughters per round of reproduction. Plus, that mother mite can then go off and repeat the cycle again. So it doesn't seem, that like, oh, 1 to 2 is not that many offspring. But when we think about the bigger scheme of things, and these foundress mites being able to do this life cycle, or the females in general being able to do this life cycle 3 to 4 times, that's when we start getting into really high numbers of these mites. And I mentioned this because, and I made a simple model for us to think about and look at, because a lot of times beekeepers, I'm guilty of this too, we get busy with life, and then we don't check our bees as often as we should. And so we'll monitor for our mites, and then the next time we go out and monitor for mites, it can be shocking how quickly those mite numbers can have increased over time. So I'm creating here a very, very simplified model, but it makes the point that I think needs to be brought up here, and how quickly these mites can reproduce. So typically we talk about most of the mites being in the brood, and that reproducing in those brood cells, and then 25% being on the adult bees. And that number does change. Sometimes people will say 60% are in the brood. Sometimes people say 80% are in the brood. So just for simple math, I made it 75% are in the brood and 25% on the adult bees. So when we talk about worker brood, so female worker bees, they, I just showed you that life cycle. They usually have 1 to 2 offspring per foundress mite. So that means I just averaged here about 1.5 offspring per reproductive cycle. So now if we look at the number of mites that are on the adult bees, so that 25% and then 75% in the brood, if we start out with a population where we have 1 mite to 3 mites, so 25% on the adult bees, 75% in the brood, the next round of reproduction here, assuming that we have 1.5 offspring per mite, when we do this, then that means all of these mites that are reproducing in the brood have 1.5 offspring plus then those original mites. So now we went from 3 mites to 7.5 mites. And that's just within approximately a little less than 3 weeks right there. And then if we do the math to figure out how many are on that adult, when we do our sampling, it's 1 to 3, or 1 part on the adult bees and 3 parts on the in the brood. And then we look even like 3 weeks later after that, when we look at reproduction, now you can see our numbers have increased even more. So now we're about almost 19 mites in the brood. And then when we do our mite monitoring, we'd have a little over 6 here. And so you can see already how quickly you can have those mite numbers creep up in just a month and a half, or about a month. But then what's even scarier is when we look at drone broods, so male bees, they take longer to reproduce or to develop. And because they take longer to develop, that means more of those mites are able to mature in the brood cells. So they have I'm saying an average of about 4 offspring per mite. Some of the literature says it's even higher than that, that you can have even 5. So I'm just being a little bit conservative here and saying that you have 4 offspring per one of these mites. When we look at the same type of model here, so here's our male bee, here's the drone brood. So for those of you who are beginners, it actually looks different from the worker brood. It's puffier and it's a larger diameter cell because the male bees are larger. And the male bees you would not see outside foraging. Those are only the female worker bees that are out visiting flowers. The male bees you only see outside on mating flights or in the colony itself. And so again, if we do this math, we start out with 1 to 3, because of this higher rate of reproduction in drone brood, because of the longer development time, um, here we are with the higher number of offspring being able to be produced. And this is a little more like 3 to 4 weeks because it takes longer for these males to develop. But then you can see more quickly just by the second. So this is one round of reproduction. The second round of reproduction now, we're getting up to 75 mites from starting from that 2 generations ago. So we have a high number of mites that can easily start reproducing in those colonies depending on what type of brood you have in there. Typically in the spring, you'll have more drone brood, and that's what leads to really high numbers then that we see in the summer. It's because the drone brood is there earlier in the season. So this is just a reminder that our numbers can get out of control really quickly if we are not managing or treating for these parasitic mites. Another problem is that while these mites are feeding on the developing bees as well as the adult bees, they're spreading viruses. And so this is one of the most common viruses that we're dealing with. It's called deformed wing virus. And so the bees that have been fed on during development emerge as adults with these little shrunken wings, so they can't fly. They're also full of virus themselves, so they can spread virus to the other bees in the colony as well. And here on the right-hand side is a worker bee, and then this other bee on the left-hand side, this is actually a drone. This is one of those male bees. And so while he doesn't do any work, so you can imagine this worker bee not being able to fly. That's a huge loss for the colony because she can't bring back resources. But this male bee, if he can't fly, he can't go and mate with virgin queens. So that's a huge loss as well in terms of spreading genetics and having successful matings. So this is a problem in multiple respects. So really problematic that we have parasitic mites that are feeding on our developing bees, making them weaker. A lot of the research also shows that they have shorter lifespans. There's just a whole list of issues from these parasitic mites, but also this double whammy of them spreading these viruses. So how are we doing and what does it look like in terms of our mite levels across the United States? We fortunately have a honeybee survey that's done by the USDA as well as the Bee Informed Partnership. And so I'll go through these charts, but I don't want to spend too much time on them. I just wanted to show you what our levels look like across the season for these parasitic mites. And so we're typically sampling 300 bees, and then we count the percentage of infestation. So then we end up counting the number of mites per 100 bees. But if you just sampled 100 bees, it's not a large enough sample size. That's why we use 300, and then we calculate the percentage. And so this time of year, we have relatively low numbers, but as that season goes on, you can see these blue bars that they're getting higher. Actually, 2015, we won't pay too much attention to just 2015. Just look at this black line that's showing multiple years of like over a decade of data here. And you can see that it will start increasing in the summertime, and then it'll start to decrease and start crash down in November and December. And so I wanted to show you 2015 because it's about 5 or so years ago. And this red line here, this is typically our economic threshold, where we usually treat if our mite level is above 2 or 3%, depending on where we are in the season. So I just put it in there at 3%, but in other parts of the season, it's around 2%. And that's where we would make sure that we're intervening and doing some type of a treatment. So if we look at 2019, I pulled up the data for that. And at first I thought, oh, that's great. Because just, flipping through, you can see, oh, it's gone down. Like we're doing a lot better. But when you look at them side by side, it's because the way that they presented the data, the axes are different. And so it's really it's a little bit deceptive here. And so if you actually stretch that graph on the right hand side to match the level of the axes on the 2015, you can see that it's really not looking that different. Um, our average is looking about the same. 2019, the blue bars, it's not very different from the average. So we aren't, we have the opportunity to still do a lot more improvement in how we manage for our mites. So we don't have them under control. We want to see this curve go down. Obviously, we want it to be below the economic threshold um, for more of the season than this. This is very problematic when we get into the part of the season where we should be preparing them for the winter. That's when they're actually dealing with their highest level of parasites, and that's not good in terms of winter survival. So speaking of coming out of winter, that's where we are right now. And this is something that I'm preparing to get a number of calls. And Chris and Scott, I hope you're listening because you're probably going to get some of these emails too. If you start seeing pictures like this getting submitted with all of these bees, and these are all dead bees, even the ones that are look like they're lively on here. But when you start seeing a frame that has a bunch of bees with their butts sticking out, so their head in, and they're dead and they're in those cells, this is a sign that this colony starved. And so you can also see there's no honey or nectar around this. They started rearing brood, but they ran out of food. Not only food to keep the brood warm, but they didn't even have enough honey to keep themselves warm. And so this it's a mix of starvation and freezing. If they don't have enough food, they can't warm up their bodies enough to stay warm through the winter, and so they end up freezing. So if you start seeing a bunch of butts in the cells, that's because the bees froze out. And so this is from one of my colonies. And so I'm not afraid to tell you guys, yes, that I have losses too. And I'll point out though that my losses are usually tied to some other issues. So we're gonna zoom in a little bit closer here, but hopefully you all see that this is not a good sign when you see all these bees packed in. It's a terrible way that they die. So, what do we see here? So we can already see that there's this one dead bee hanging on to the outside, and then we have these little see these little bee rear ends sticking out in the cell. So these are all dead in there too. So what do we see here? Do we see anything striking other than dead bees? Because this is the question that we often get: is why did my bees die? And so I challenge everyone to take a closer look and be careful when you're pulling apart your dead colony and your dead frames, because I treat it like a crime scene and that we have to try to figure out what's going on. And so you don't want to automatically be too rough with the frames and have all the bees fall off, because seeing the bees themselves on the frame and where they are can actually help provide some clues to what happened. So we're going to take a closer look at this bee, just a slightly different angle and closer up. And what you can see here is between her thorax and her abdomen, there's a parasitic mite. It's also dead. It didn't make it through the season either. It's also frozen on this bee. But this is a sign that this colony had mite issues. So that's probably one of the reasons the colony population was so small that it couldn't keep itself warm, even if sometimes even if they had tons of honey in there. If the population of bees is so small and it gets really cold, they cluster so tightly together that they won't even go an inch or two away where there is honey and nectar for them to eat. So sometimes it'll be deceiving when you look at a colony and they say it's heavy, they have honey in there, but they still died of starvation. It's because oftentimes those populations are too small and they can't access the actual honey that's in there. So those are really sad cases because they had food, they just weren't strong enough to actually get to the food. So again, pay really close attention and look at your dead bees to try to figure out what could have gone on in there. And they have these little pesky places where they like to hide out, and so you got to look pretty closely. Another thing too is that when I have a dead colony, I look at the bees that fall to the bottom of that colony. So I have screen bottom boards on my colonies rather than solid wood. And I look at those bees. I try not to rustle them around too much because you can get things to fall off. But then what I do is I try to take pictures. I try to look at them in the time point, but then I'll also take pictures so that I can look at them closer up on my computer later on. And so what we see here is there's a mite, there's a mite, there's a mite, So it's very problematic that just within the small number of dead bees here, there are dead mites. And there's probably some dead mites that have fallen through the screen as well and that are underneath. But this is a sign that my mites were not under control and likely contributed to these bees not making it through the winter. So as much as I'm pointing out here to look for these dead or look for these mites on dead bees, I also want to point out that when we're dealing with live colonies, we do not trust visual estimates of varroa mites. So it's really easy for us to look at them on dead bees, oftentimes because they're flipped over and you're seeing all different parts of them. But with live bees, we're usually not looking on the at the right parts of the bees, and we'll get to that in just a second. I forgot that I have another one to show you. So this is another dead colony. So you can see that they clustered so tightly together because they were trying to stay warm. Here you can see one of the queen from this colony, which is marked yellow, to help find her. But she's obviously really easy to find in a small population like this. But again, taking a closer look at these dead bees and trying to figure out what happened, there are 5 mites on just this handful of bees that are more than a handful, like a literal handful, the small population of bees here. So mites were out of control. And hopefully, as a beekeeper, you should not have mite levels like I do. My mite levels are not, I wouldn't say artificially high, but I maintain high mite levels for research projects. So as a beekeeper, you should have your mites under much better control than I do for my purposes. But it's if you have losses, don't think that you're alone. You have room to improve every year. So don't feel discouraged and feel like you can't do it. It's just that you need to adjust your management for the next year to be more successful. And that's what we're here to help you with. And so again, we can see these dead bees and we can see the mites on these dead bees. But when we talk about live bees, it's not that we look at the live bees and count the mites the same way. And that is because on live bees, these mites tend to prefer, and you don't have to worry about all of this. If you're a beekeeper, you might be more interested in looking up the study. But if you're someone who's thinking about getting into bees or early on, just want to make the point that the bee's favorite place to be is on the underside of the bee. And typically, when you're looking at bees, they're not showing you their bellies; they're showing you their backs. And so if you start seeing them, like I showed you in that first video when I started out the presentation, where there were a lot of the mites visible on the thorax of the bee, only like 1% of the mites that is their preference place. So if you're seeing a lot of the bees that have the mites there, that means there's probably a whole lot of mites on the undersides of the bees that you aren't seeing easily. So that's why we don't trust this visual estimate, or if you are seeing them easily on the backs of the bees, your mite levels are really high. And so we want to make sure that we're catching it before it gets really high. So we have monitoring methods, but just remember that the mite's favorite place is not on the back of the bee, it's on the underside of the bee. And that is not where we're typically looking when we're looking at bees. Um, they don't like to show us that side of them. So we have monitoring methods, and this is a whole separate talk and a session that we do in the Master Beekeeping program. So I encourage you to take them to the Master Beekeeping program. We'll have them this spring. I think we're gonna have to do it virtual, but we can get into that a little bit later. But just pointing out that we have some really easy ways that we can test and monitor for parasitic mites, these varroa mites, and they aren't perfect, but they are definitely more helpful and more accurate than just looking for adults or just looking for mites on adult bees. So one of them we're using isopropyl alcohol, which I know is hard to get your hands on right now. So actually, I'm working on a research project to find some alternatives. And this one, you destructively sample your bees. So you take 300 bees and you put them through an alcohol wash and you shake them up. And that dislodges those mites off of the bees. So it gets them out from those little nooks and crannies of the bees. And then those mites will then drop down. And then you count the number of mites per 300 bees and then divide it by 3, and then you get the percentage of mite infestation. Same thing with the powdered sugar roll, except it's slightly different. The bees are alive. It's not destructive sampling. You use live bees, and you can release them back into the colony afterwards. A lot of people like that will oftentimes not shake the bees hard enough to get an accurate count, because a lot of people that like this or like this method don't want to kill the bees. And because of that, heart helpfulness or the good intent of not wanting to hurt the bees, they oftentimes don't shake them hard enough. And because of that, then your mite drop numbers can actually get can be a little bit inaccurate. So if you're going to do powdered sugar roll, I encourage you to make sure that you are doing it as effectively as possible, because all of these monitoring methods are only as good as your ability to conduct them. So for me, I actually prefer doing the alcohol wash because it is a more accurate count. And it's faster for me to do those than the powdered sugar rolls. It's also hard in the southeast. We have a lot, depending on where you are, you can have really high humidity on certain days. And when you do that and you do powdered sugar, the bees can get kind of caked with powdered sugar. And again, then you don't have an accurate mite count. But this is a whole separate talk, like I mentioned before. We spend an hour just talking about bees. So I don't want to spend too much time on this because we can talk about it more in the future. They're also using sticky boards. So we put these boards that are sprayed with nonstick spray. It's ironic that we call them sticky boards, but we spray them with a little bit of oil and we put them underneath that screen bottom board. And that allows us to count the number of mites that drop down off of the bees when they're grooming, or sometimes the mites just happen to fall off. And that gives us a little bit of an estimate of the number of mites that are on the bees themselves. And then lastly, we have or not lastly, this is another method. There are others. I was looking at our drone brood. So I started out, remember, showing you how quickly the mites can reproduce in drone brood. So we can actually look at drone brood, since the mites prefer them, and pull those drone brood, the drone larvae and pupae out, and look for mites on them. We don't have good thresholds though for either sticky boards or drone brood. So oftentimes when we're talking about monitoring for mites, we're looking at both alcohol wash and powdered sugar roll mainly. But I do encourage everyone to be on the lookout. And so I do alcohol washes. Sometimes I'm doing powdered sugar roll depending on the experiment, but I'm also on the lookout. So when I do have drone brood, that's the other issue with drone brood, is you don't have it available throughout the year. You typically don't have much drone brood in the late summer and fall. And that's when your mite populations are really starting to go up too. So that makes it a little bit difficult. But when I do have it, and bees tend to build this extra burr comb in between the boxes, and they'll oftentimes build drone brood in between. And so I'm very quick and ready to go when I'm pulling my boxes apart to look for that drone brood and to see if there are mites reproducing in that burr comb in between. And oftentimes you can find evidence of the mites reproducing there just within those cells. So here I can already see at least 3 mites there. And so that's too many mites, and so that's a problem, and is a sign that I need to start getting on top of my management. The other thing too is just popping open some of those cells can be helpful and informative, and seeing whether or not you have mites in there and what the mites are doing. Do you just see one mites, or in the case of some of my colonies, I'm looking and I'm finding whole families of mites. So here you can see that foundress mite, the mom, that's this dark reddish brown, but she started her family in here. So here are her babies. This is their little waste pile. So you'll oftentimes when you open those cells, that's the first thing you notice are these little piles of white waste before you even see the mites, because of the color difference there. So this is a very bad sign. It's one thing to see the mites. It's another thing to see them reproducing. I consider that like stage 4. If you were talking about cancer, this would be like it, because what else are you waiting for these mites to do? Their populations are increasing, and this is like your last chance to really get to do something about it. Okay, so kind of going with the theme of murder hornets and murder mites, I just wanted to point out to you, because I can show you all this, and we're oftentimes looking at these mites on bees and stagnant or in the cells, but I want to show you a quick clip that I made, just showing you how much these mites love honeybees. And it's meant to gross you out a little bit, but also to make the point that you have to be on top of your mites, because they are on top of your bees. So this is a poor dead bee, and these are some mites that I shook off from some other bees. And just trying to make the point here that these mites are very well adapted and very well evolved to attack honeybees. And of course, that being on a paper towel is not very attractive to them, but they are able to detect their host honeybees, and they will in a heartbeat, or less than, they don't have quite heartbeat, in less than a skip of a heartbeat, they will take the opportunity to get onto a honeybee because that is their host and that is their chance for survival. And so I want us to think about these mites, and I want you to be grossed out by this, and I want you to remember this, that these mites are going to your bees like a magnet. And so you really, really need to be fighting them off, and protecting your bees, because right now they are winning the war essentially. Oops. See. Oh, here's a little bit of a close-up video of it. Let's see if that one will play. And just showing how when the bee gets close up to, let me see, maybe I need to stop this one. When the bee gets close up to the mite, that mite doesn't, it just immediately will just start climbing up that bee, because this is her chance for survival. That mite has to have these bees in order to live or reproduce. They don't have other hosts. They need to feed on these developing bees. And so be grossed out. Make sure that you're keeping your mites under control because they will take over your colony. Okay. And just remember too that we have to be on the lookout and think about being responsible beekeepers. So these bees are also, if we're doing, we have to be careful about things like bulk feeding. So in some areas where I've lived in a number of states. Bulk feeding is a little bit easier than other areas. Where I am now, I have too many other beekeepers around me, and so I don't recommend bulk feeding out in the middle of the apiary, because I'm attracting bees from other apiaries as well. And if they haven't treated their mites, some of the bees that come into my apiary have mites. And because these mites are really quick and they can transfer from one bee to another rather quickly, that's an easy way to transmit these parasitic mites and those viruses from someone else's apiary into mine. And so it's this reminder that what we do in our apiary affects the beekeepers that are also around us. And so now I just have a really short video to show you, just showing you how fast varroa mites are. And so this is, again, like, do as I say, not as I do. This is from one of my colonies that is highly infested with mites. And so you can actually see here, there are 2 mites on this bee. So she has a double decker, a short stack of mites on her. But I'm going to tell you when one of these mites transfers to another bee, and it happens in a split second. So I just want you to think about how quick and how efficient these mites are. So you're going to see a bee, and you're going to see not lots, but you're going to see several bees with mites on them. But we're going to see a bee come over from the right-hand side, and she's just going to quickly make contact with that bee that has the double stack, which is now looks like one stack. Let's see, it's this bee here on the upper right. I'll say beep when the transfer happens. Beep. So it just happened. So now there's only one mite there. I know it's hard to tell. There's only one mite, but we wait a second, and you can see now that's the other mite has already gone on to this bee here. So all it took was that short moment of close contact between those 2 bees for that mite to be opportunistic and jump onto another bee. And so this is just such a, I mean, it's shocking, but at the same time, it makes sense when you think about how well these mites are doing in our honeybee colonies. And so we really, really need to pay better attention to what we're doing and how we manage these mites, because they're very successful. And I don't want to say that they're winning, because I don't like to think of them that way. I like to think that we're still doing our part to fight them, but they are a bigger challenge than I think we had anticipated, and that some beekeepers, I think, treat them to be. They are a major problem, and we can't let them go without attention. Okay, let's see. So what do we do, right? So that leads us to, like, okay, hopefully I've convinced you that these are murdering our bees. So what do we do about it? So managing varroa mites is key. And so we can talk about integrated pest management. And so we have different types of management methods, and different levels of treatments. And so we do have chemicals that we can use. We have behavior. So we have selected stocks that are more resilient to parasitic mites. They're not a silver bullet. You still have to use other methods as well. One behavior, though, is varroa sensitive hygiene. So these bees are really hygienic about detecting mite infested brood cells, and they'll uncap and pull out the developing bees. And then there are also bees that are really good groomers. So when it has a mite on its body, it will try to groom it off of its body, and they'll oftentimes like try to bite at that mite and do some damage. And we also have some other IPM methods that are non-chemical. So things like, we know that the mites really love drone broods. We can actually try to trap them in the drone brood, attract them into the developing drones, and then once those cells get capped off and they're in there trying to reproduce, we take that entire frame out and we freeze it. And that way, none of those mites are able to reproduce. So it's one non-chemical way that we can try to deal with mites. And so in reality, we should be practicing integrated pest management where we have a whole slew of methods that we can use. And we want to be able to rotate chemicals if we are using chemicals so that we don't encourage resistance in the mites. We do have issues with resistance in some of our chemical treatments right now. And we also want to combine treatments too. So making sure that we get the most bang for our buck. So remember that most of those mites are in the brood cells reproducing. And most of the chemicals, there's only one chemical that claims that it can penetrate those cappings. So if you do use a chemical treatment, it's only affecting the mites that are on the adult bees. And that's the minority of the mites. So there are things we can do to try to get more of the mites on the adult bees so that when we do a chemical treatment, we'll be able to have a higher efficacy level. So we can talk about that if we have time during Q&A. So I just want to show you this. So this is for those of you who are non-beekeepers, this may not make a lot of sense, but for those of you who are beekeepers, hopefully when you see this, you'll see that there's a difference between the left-hand side and the right-hand side. And so beginning beekeepers may see this and think, and normally if we were doing this in a classroom, I could ask you and you guys could respond, but we're stuck in the virtual world, I'm sorry. What people would see on the right hand side, they'd say, oh, that's a spotty brood pattern. So normally when we like to have all of our brood cells, all of them capped at the same, or seeing like a really nice pattern where they're almost all capped together. Whereas on this side, you can see there's some spottiness here. These horizontal lines, those are made by the researchers, so just ignore those. But you can just see that it's a patchier shotgun pattern here. And so we oftentimes will attribute that to the queens. But in this case, it's actually Varroa sensitive hygiene. So it's actually a good sign that these bees are uncapping and trying to remove mite infested brood. It might be a bad sign that you have mites, but at least the bees that are hygienic are trying to do something about those mites. Whereas this half that was put into a control colony that was not bred for hygienic behavior, it has the same level of mites as the section of brood that's on the right hand side, but those bees didn't respond to it. So those mites are allowed to reproduce in those cells. And so this is something that we pay a lot of attention to, is trying to make sure that we think about how we're breeding our bees and trying to breed for traits that are good for their health and not just honey production and things that benefits humans, right? So we need to make sure that we're benefiting the bees and how we select for traits. So this is just a reminder that when we look at things, we're doing our inspections, if we see something like a spotty brood pattern, it may not be because you have a bad queen. It could be something like you have hygienic bees. And I just wanted to point this out for those of you who are beekeepers. There's a recent study that came out looking at this, the pattern of brood from good queens and poor queens. And then when they did a reversal and they switched them around, what they ended up seeing is actually that it wasn't really necessarily that a poor queen or a poor pattern queen was a poor pattern queen. Oftentimes those bees or those queens, when they were put into a colony that had a good brood pattern, those poor queens actually were able to maintain a much better brood pattern than they had originally before. And so there are different levels of effects of what's going on and underlying this. But I just wanted to point out that brood pattern alone is not indicative of your queen quality. So don't think that if you have a bad brood pattern that you just need to kill your queen and requeen and start over and that things will improve. It might, but it might be a more an issue that has to do with parasites or brood or hygienic behavior that doesn't necessarily warrant killing your queen. Oh, sorry, that's just some results. And then when I mentioned drone trapping before, just wanted to show those of you who are beginners of that frame that you can put in. So this is it's a green plastic frame. This is kind of the standard that we use, but you can also get them to build drone comb underneath some of your other. You can use like a shallow frame and put it in a deep box, and they'll build extra burr comb, and it'll oftentimes be drone comb. And that's another way you can get them to make this. But the key for this, so because drone brood is more attractive to the mites, this becomes kind of like a mite magnet. So your mites will go into the drone brood, and then those cells will get capped off, and then the mites will start reproducing in there. So the key here, if you're going to use this method, is to make sure that you freeze this frame before those bees start to emerge. So keeping a calendar and records is really important for beekeeping. So is otherwise, if you let these bees start to emerge out, now you've just raised a bunch of varroa mites and you've made the situation worse. So only use this drone trapping method if you're really good about being on target with schedules and calendars. And I mentioned this before, that breaking the brood cycle or combining methods, sorry. So one way that we combine the method, and I think I said I'd answer this during Q&A, so I guess I'm preemptively getting to it. If you break the brood cycle, and there's many ways to do this, is basically you're keeping your queen from laying. And if you keep your queen from laying, instead of having all these brood cells that those mites can reproduce in, if you cage the queen or you move the queen temporarily or you split your colony, now you have a situation where you have a new virgin queen, or it's going to be queenless for a while. And during that time, eventually all of your pupae are going to develop into adult bees, and they're going to emerge out, and there won't be more brood for those mites to reproduce in. So you end up having a bunch of open brood cells, and then all of the mites are going to be on the adult bees. So now when you use your chemical treatment, it's going to be much more effective. Okay, I know we're running short on time, so I'm going to skip this study. I want to show you, because now it's going to be, what do I use, right? How do I treat for mites? And so I'm going to try to skip forward to try to share, so I can show you this website. So the Honeybee Health Coalition has this really great guide. It's a pretty hefty guide, but it has information about the biology of the varroa mites, the different treatment methods, IPM, as well as chemical and non-chemical methods. Um, and they also have this decision tool, which I think is what you all would be really interested in looking at. So let me pull up that page. I have too many pages open. It's not showing. Oh, it should be on Safari. Let's see if it will. I don't see any of my Safari options here. I haven't had this problem before. That's always fun when it's something new. All right, we'll try it. Oh, wait. Oh, now it popped up. It's playing games on me. Okay, so here's this decision tool. And so it makes it really easy, and it has a lot of helpful information too. Even if you get to a question, you're like, I'm not really sure. It has information to help you. So, okay, we're going to hit get started, and it gives you some background information. We're just going to click through this because of time. So Chris, I think you'd sent me an email, and I'm basically going to tell you that this is the site you should go to send to this beekeeper, but this will be a great way for me to show you. So if we look at this, they'll ask you a series of 5 questions. And so after sampling, assuming that you're monitoring for mites, did your mite level exceed the treatment threshold? If you don't know about treatment thresholds, you can then click and it'll give you information about what those thresholds are and how to do it. They also have videos on their website to show you how to measure. So we're going to say, yes, our mite levels are above. Are you open to using synthetic treatments, or do you only want to use naturally derived treatments? I am open to everything. I want every tool in my toolbox, but this is a personal choice. And so, you have to decide for yourself. But for me, I'm open to everything. Is brood present? And so we're at a tricky time of the year, depending on where you are. You may not have brood present. You may have brood present. I think where I am in some of my colonies, I do have brood present. Do I have honey supers on? I do not have honey supers on yet, right? So there's honey in the colony, but it's for the bees. It's not for human consumption. And all these questions, like whether or not you have brood and whether or not you have supers on, it's because your different chemical treatments have different restrictions. So this is helping you find, based off of your conditions, what chemicals you can use. So no, I do not have honey supers on yet. And so where are we in this population stage? We're somewhat in between dormant and increasing population, but I'd say because I have brood already in my colony, I'm going to say that I'm in an increasing population. if you're in an area where it's still cold and they aren't reproducing yet, then you would probably pick dormant. But I'm going to say I'm an increasing population. So here are all of the options for the chemical treatments I can use. And then you can click on each one of those to get more information about what the label says and how you use them, the advantages, disadvantages, all the cool information and questions that I'm sure you have about all these products, mode of action, how long that treatment is, really valuable, helpful information. There are, with everything, nothing's perfect. I do have a slight issue with some of their content. This is one of them. Formic acid is not allowed in the United States, even though they have a little flag on here that says, it is not allowed. You cannot buy formic acid as a liquid and just use that because it's not a labeled product. We have to only use registered labeled products, and that's these other ones, but not formic. And then you'll see here, some of these are temperature sensitive. So it makes it really hard in the heat of the Tennessee summer to treat for mites when you have really high temperatures, because a lot of these you won't be able to use because they're volatile and you'll have a high level of the chemical and that will actually kill some of your bees. Oxalic acid, you notice, does not have a temperature restriction, but it's not recommended for when you have developing bees, and that will be in the spring and summer. So that's also complicated. So again, this is why I don't have one chemical that I always go to. I don't have a simple recommendation. It's complicated, and that's why I like showing you guys this tool so you see what those options are. Okay, we're going to quickly go and finish the PowerPoint. Okay. Oh, wait, we already did that. Okay, so I want to make sure that it's very clear that you monitor both before and after you do treatment, ideally, right? So you're monitoring beforehand to make sure you need to treat, because there's no point in wasting money on a chemical treatment if you don't need to treat. And also that can encourage development of resistance. So we want to make sure that we're only treating when we need to and making sure we've reached that threshold. But then we also want to treat afterwards to make sure the chemical treatment worked, right? Because if it didn't work, you can't assume that it worked, because unfortunately, there are a number of times when it does not work effectively. And so you need to know that so you can then follow up with another type of treatment. So make sure that you're monitoring beforehand and that your treatment actually worked. This is just showing those sticky boards, so different ways that we can monitor for mites. And just remember that what you do affects your neighboring beekeepers. So since you guys are all part of a community and a group, be thoughtful of those that are around you. And I know a lot of people say, oh, it's just me and my bee yard, and it's no one else's business. But your bees fly and they can spread disease rather easily and quickly. And so we really want to make sure that we're being responsible beekeepers on top of being successful beekeepers. And I just want to point out too that there has been a recent study. I know I'm talking about all these chemicals and breeding, and that's kind of far off. There's a recent study just showing some basic management that can affect. Here we're going to just focus on the mite loads and the drifting here. Honey production is still important, of course, but just how you set up your apiary can affect the levels of mites and then the drifting. So bees accidentally going from their colony and then drifting into another colony by accident. And when they drift, they can also spread disease that way. That's why we're concerned about it. So, we typically set up our colonies. They are one color. They're all facing the same direction. They're rather closely together because that's convenient for us. And so what they did is they actually set up their colonies in a different way, where they have them at different heights. They have them at different colors. They set them up in a circle so that they're all facing different directions. And they're 10 meters apart as opposed to 1 meter apart. And they saw a significant effects in terms of having lower mite levels and lower drifting when you set up an apiary like in this round circle as opposed to in a straight line. So that's just something really simple, circle and spaced out, and uniquely identification, but also the colors and the uniqueness of those colonies can have an impact on your disease levels. And that's something so easy that we can all be doing. And so I encourage you to think, kind of outside the box of what we typically do, and think about what we should be doing and not just what's convenient for us as beekeepers. I'm going to skip through that. That's just robbing behavior. So they'll steal from each other, and that's what it's bad. They'll steal honey from one another, and that's no good. So just kind of lastly, touching on the importance of record keeping. Scott and Chris probably work with a lot of other stakeholders that are much better record keepers than beekeepers. We tend not to be the best at record keeping. I think maybe because we love our bees so much, we get so enthralled when we have that hive open that we don't want to be taking notes. We just want to be like there with the bees. But we really need to be taking notes on what we're seeing so that when we do get to a point where we start seeing disease problems or low colony numbers, that we can then, or hive populations, we can then track back in our records to try to figure out how we got to that point and not just rely on our memories because we don't check them often or regularly enough to really know. So I always remind myself that in the moment, I hate keeping records because it feels like I have other things I should be doing, but those records are for the future me when I need to look back. And so think about that you're helping yourself out for the future. And know that you're paying attention to your bees, and we can talk more about actual record keeping. But you're also paying attention to your management practices because maybe you end up having a really bad year or good year, and you need to know why that is so that you can replicate or change your management for the following year, and really paying attention to how you have everything set up around your apiary as well. Sorry, I already showed that one. And so I just kind of want to leave you with a little bit of a positive note. I know, we didn't talk about murder, but I want you guys to think too about how important it is that we have this great organism that we're working with. They're super amazing. They're super helpful in terms of pollination services. They're fascinating. And that what you do as a beekeeper can have cascading effects on the other beekeepers around you. And it's normal to make mistakes. If you're a beginner or if you're experienced, you're going to make mistakes, and that's fine and that's normal. It's what you do about those mistakes that really determines what type of beekeeper you are. And so we have the opportunity to improve each year, and we're coming on a new year. So I hope that everyone sets high goals to be the best beekeeper they can, not only for yourself, but for the beekeepers that are around you. And so I want to thank you all for your time and your commitment to bees and learning more about agriculture and putting up with the virtual education world. I have a website. This is the short email ad, or the short URL for it is just tiny.utk.edu/apiculture, or I'm sure Scott or Chris can share it with you. Or I can put it in the chat box as well. But yeah, please, 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 if you have questions about bees, the way we have the structure set up now, I can harp on you guys since you're here, is that Scott and Chris are your go-to folks. And then if they can't answer the questions, then they send them to me and then I answer them back. And so that way they learn the answers too. So we're really trying to build this whole network in Tennessee because we have this explosion of interest in bees and beekeeping. I can't handle them all of them myself. So we have these great extension agents all across the state that are learning about bees. And I think Scott even has bees now, right? So they're one of the community. And so we're really working hard to create an entire network where we can assist you in a more timely fashion and also in a more local level. And so thank you guys so much for your time. And so I guess I'm not sure who the question wrangler is, but if there are any questions, I'd be more than happy to try to answer them.
Dr. Tsuruda, I want to thank you for doing such a good job. As these questions, there was a few questions came across on chat and stuff. You answered a lot of those as you went through.
Oh,
one
good.
for sure. Don Kimball had a question here. It says, what if you have a clean hive that you just installed? Where do the mites come from? And just automatically you led right into that with talking about the drift from neighboring colonies and all that. And I guess that's where that would come from in that case.
Part of it. And then another part of it is that they come with the bees themselves. So you'll oftentimes get, marketing is one thing, and people say, oh, I'm selling you Varroa free bees, or they're hygienic bees, and that's what stock they come from. But there's no like pure stock of bees that I have a whole lot of reliance on in terms of saying, oh, those are your go-tos and you'll be okay with them. So yeah, they'll come from other bees that are around. They'll come throughout the season, later on in the season. They'll rob from the weaker colonies and get diseases that way too, or get parasites and disease that way. Yeah, unfortunately. Any other questions I need to address? I can't tell if Scott is really still or if he's.
It looks like he froze. It looks like he froze.
I was like, he's like really bored. He didn't like my answer. Let's see. Okay,
let's see. We got in the question: can mites actually fly from hive to hive? So it sounds like it's more likely that they're flying on the bees. Is that correct? Rather than the mites flying?
So yeah, mites actually they don't have wings, so they can't fly. They hitch a ride on the bees. So that's why they're so tightly intertwined with how the bees live. They're reliant upon the bees, and so yeah, they can hitch a ride and make a transfer out of flower. They can make a transfer when one bee is robbing out another colony. They're really resourceful in that way of spreading. But yeah, they can't fly themselves and they can't walk very far. They're really small. They can't walk very far. So they're using the bees, unfortunately.
Got a question here. How often do you open a hive to check your bees?
So it depends on the season. This is the thing that when you ask about bees, there's a lot of it depends, and so this is one of those where it's perfect of it depends. So obviously, this time of year, not doing much inspecting, especially with the cold air, you don't want to be interfering too much. But I do check when it is warm enough. So like last week, we had a day that was or a couple days that were the 60s, and it just happened to align with my schedule that I could go out and check the bees. And so I went out and checked on them. Some of them are doing really well. Some of them are not doing very well, or might need a little bit of more help. But then come spring, some of them, I'm already worried they're going to start swarming soon. I can tell they're very strong for this time of year. And so they're going to start swarming soon. And during swarm season, I'm checking them about every week because it takes about 16 days for them to make a new queen. And if you don't catch it early on, then you're going to lose at least half of your bees in that old queen, and they're going to end up somewhere. And as a responsible beekeeper, you don't want them ending up in your neighbor's walls of their house or wherever else. That leaves a bad taste in non-beekeepers' mouth. And so we need to be really responsible about that. So I do a lot of swarm control, and that's what I'm going in and checking every 7 to 10 days. I like to say 7 days, but life happens, and so it ends up 7 to 10 days, and making sure that I'm cutting those queen cells and making sure I'm doing what I can to control those swarms. Once swarm season ends, I can relax a little bit more and not check them as often, but I try to check them ideally every 2 weeks. But it is tough when you have other things that happen in life. And I mean, working from home or the shutdown has made things a little bit easier, but the workload still kind of gets in the way of checking bees when you need to, or the weather.
Cindy asked a question we get a lot. Where can you get good bees from? I know that may be a loaded question, Dr. Tsuruda, but
Yeah. So I would ask in your area, so you have all these local clubs. I would ask your local clubs where where people are happy with getting their bees. So we do, we have this issue where we have a lot of bee producers and a lot of it's coming out of southern Georgia. And so you may want to find out too of who you're buying from, whether they're the actual producer or if they're the middleman, right? Because if they are the actual producer and the middleman, they may not have information on where they're actually coming from. They should. And know too that if you're getting something from out of state, that if it's on comb, you have to have a permit for entry into Tennessee. If you're buying packages right now, they say that you don't have to have a permit, but it all just depends on TDA's lawyers. And when they switch lawyers, they can interpret the law a different way. So always pay attention to the TDA website and keep current with that. But also keep in mind, if you're starting with a package, which most people are, so it's like 3 pounds of bees and there's a queen in a cage in there, I consider that a starter kit. And so I will use that, but then I will put in my own queens that I rear later on. So I use that just to get the ball rolling. But then over time, there's a turnover in the bees with the stock that's in there. I want to make sure it's one of the stocks that I want and not something that's been bred in a different region under a different climate. So yeah, sorry, I don't have a good just go buy it from so and so. But keep in mind too that you can buy your starter kit from so and so and then put in a queen from someone that you have paid a little bit more investment into in terms of knowing what they're doing and that they're breeding good stock.
we get a lot of good comments here. People enjoyed the session. Got a question about master beekeeping, where they can find those dates. I'm sure that tiny.utk.edu/apiculture, when you have those dates, that's where it'll be.
Yeah, so it's unfortunately, and I guess fortunately, it's looking like I didn't want to do it this way, but it's going to be virtual this year. And so because we just can't rely on health and safety at this point. So let me put my website. I put it in the chat. So right now, I think it says that, you know, we're working on it, which is, it's true. We are working on it. So I think we've decided, the working group has decided that we're going to go virtual. But as far as like picking what it'll be in the evenings. And so I have to get approval from TDA at this point because we're changing the structure of the class. And so that's the thing I'm waiting for right now, is making sure that they're okay with it to give the certification off this new system. And so, you know, realizing that there are going to be issues with certain nights people may not be able to make it. We're probably going to have, you know, like 10 to 12 evenings, and you have to come to 8 to 10 of them to get your certificate. Not everyone cares about the certificate at this point if you're a beginner, and that's fine. But if you want the certificate, we're going to have because there's a certain number of hours you have to put in. But yeah, it'll be virtual, and we're going to try and make it as fun and interactive as we can. But yeah, at some point, you know, we need to get in the bee yard. People don't get into bees just to listen about bees. They want to be in their bees. And so we'll get to that point soon, I hope. Any other questions I need to address?
I was trying to look over the chat list there and got one. Given the life cycle of mites, it seems that repeat treatment is necessary to kill each new batch as they hatch out. So would you recommend multiple treatments, like 3 treatments in a row, separated a little bit of time apart, or what would you recommend in that situation?
So again, it depends on the treatment that you use. So like Apivar is the synthetic treatment. The active ingredient in that is amitraz. And so that's a slow release plastic strip that you put in the colony. So that treatment is 42 to 56 days. And it's slow release because it's meant to cover 2 rounds of bee emergence. So that way you're getting theoretically all of the mites that are in that colony because of that time period. Whereas some of the softer chemicals, they are flash treatments. So for those, you do have to do multiple treatments. That is complicated. And a lot of people think, oh, because they're softer chemicals, they're safer for the bees. But what we're seeing now with a lot of the research is that people are doing so many treatments and doing them so close together that they're actually causing some damage to the bees. And actually, one of the studies that let me see if I can pull it up really quickly, that I meant to show you earlier on, and I was just worried about time. They actually looked at one of the soft chemicals that everybody loves to use, and it's oxalic acid. And that's something I'm seeing, and I don't want to sound negative, but when I did the master beekeeping class, my first year here, 2019, we would go out to the apiaries, to an apiary at whatever county we were in. And when we would go out and we'd sample the colonies, either people were not following best management practices, or they're using oxalic acid that's not labeled for Varroa mite treatment, which is a federal regulation no. You have to use labeled products that are labeled for Varroa mite treatment. Otherwise, it's an illegal treatment. But also, we want to think about, you know, using the right chemicals that are labeled because of their use in a food producing animal. So you don't want to use off-label chemicals in a colony where you're going to be taking honey off of it. That's very problematic. And so again, we want to pay attention to the labels. But so a lot of people are using oxalic acid and they are not monitoring. And so you need to monitor before and after. And so a lot of people weren't monitoring before. And when I went to the class, we would go and we'd monitor, and their mite levels were really high. And so it wasn't working, but I can't tell if it was helping even at all because we didn't have pre-treatment levels to even compare to. But mite infestation levels that were above 12%, that's, you know, we're talking usually a threshold of 2 to 3%. So even after multiple rounds of treatment with oxalic acid, their levels were really high. And I'll point out again that oxalic acid is not meant for use when you have brood developing bees in the colony. It's meant more as like a wintertime treatment where you don't have developing bees. But a lot of beekeepers are using it during the season when there are developing bees, and that because it doesn't penetrate those cappings, it's problematic. And so just want to show you this study. They used these are different treatment levels. They used it in many different ways, even trying to interrupt the brood so that more of those mites were on the adult bees. And what they ended up finding is overall, and their control, which is Apivar, which is the synthetic, and I know a lot of people don't want to use synthetics, but that did have the best control over all of the other ways that they tried to use oxalic acid. I'm not saying oxalic acid is is not going to work. It's just the conditions at which it works. You have to really pay attention to, and you have to make sure that you're monitoring afterwards to see whether or not it worked. Because assuming it worked, you're going to get into some trouble in the fall, and then some really low overwintering levels. So there are issues with Apivar too. I don't want you to think that I'm just saying use Apivar. There are issues with Apivar. There are some growing concerns that there could be some resistance popping up in our populations as well. So we don't have a simple go-to there in terms of recommendations, but that's also why we want to rotate our chemicals. So, I mean, one advantage of the Apivar is that it is slow release, and so you put your treatments in, and you don't have to worry for 2 months. But at the same time, you can't use that chemical when you have honey supers on, so that makes things complicated. And you can't wait too long in the season to treat. If you wait until October to treat, your mite levels are too high. So yeah, it's not easy. That's why if you use that guide, that's helpful in terms of trying to figure out what to use when, and looking at all of the considerations and factors.
I think that management tool, that's going to be one that everybody's going to be referring to help answer a lot of those questions. And we do have some other questions coming on board, but in essence of time, I think at this point, I'd like to just remind everyone that they have an extension agent in each county. If they have questions, reach out to their agent. If they don't know the answer to that question, just as you'd said, we can reach out to the specialist, and Dr. Tsuruda she'll be helping answer all those questions and stuff for us. So reach out and work through your extension agent in each county.